This is Jim Schnabel doing lecture number 5 in a series of human pathology by Robbins and Cotran, 9th edition. Today we'll be discussing genetic factors and pathologic disease. Mutations are common and they're usually repaired, and these cause 50% of spontaneous abortions which harbor a chromosomal abnormality. There are 3 broad types. There's a single gene mutations with large effects, and these have a Mendelian pattern. Number 2 is the chromosomal disorders. These are structural alterations in autosomes and sex chromosomes, and they have high penetrance. And the third type is complex multigenic disorders. The polymorphous ones have multiple genes involved. Each contributes a fraction of the disease, and there is low penetrance. Mutation types include point mutations within a coding sequence, which codes for a different amino acid. These are called missense mutations, or a stop codon is produced, and this is a nonsense mutation. Point mutations can also occur within a non-coding sequence. They can either reduce or increase production of a substance. There are deletions and insertions. A shift of 3 bases shifts an amino acid reading. Others are frame shift mutations, and these are non-multiples of 3. Trinucleotide repeat mutations are situations where long nucleotide sequences are amplified. Familial or hereditary mutations, the sequence is transmitted from the parental germline. And finally, congenital mutations, the sequence is manifested from birth. Some genetic diseases are manifested later in life, such as Huntington's chorea. Mendelian disorders. We all carry a small number of deleterious genes, and these are usually autosomal recessive. Single gene mutations are often autosomal dominant, and they are manifested in a heterogeneous state, so that one parent is usually affected. If no parent is affected, then a new mutation has occurred. Penetrance is where the expressed trait is present in the genome, graded from 0 to 100%. And manifestations of an autosomal dominant inheritance often depend on whether compensatory situations exist. Among autosomal dominant disorders, famous ones are Huntington's disease, neurofibromatosis, polycystic kidney disease, familial polyposis coli, von Willebrand's disease, Marfan syndrome, Ehlers-Danlos syndrome, osteogenesis imperfecta, familial hypercholesterolemia, and acute intermittent porphyria. Autosomal recessive conditions are more common than autosomal dominant disorders, and they require mutations in both alleles. Siblings have a 25% probability of having the trait. Complete penetrance is common, and most of these are enzyme coding mutations. Among these are cystic fibrosis, phenylketonuria, galactosemia, lysosomal storage diseases, alpha-1 antitrypsin deficiency, Wilson's disease, glycogen storage disorders, sickle cell anemia, thalassemia, congenital adrenal hyperplasia, again some variants of Ehlers-Danlos syndrome, and Friedreich's ataxia. X-linked disorders. All sex-linked disorders are X-linked, and they're autosomal recessive. Y-linked mutations affect males, and they cause infertility, so there are no such things as Y-linked mutations. In X-linked mutations, sons are affected and daughters are carriers. Heterozygous females can express the trait partially. One X-linked autosomal dominant disease exists affecting 1/2 of daughters and sons from an affected mother, and all daughters and no sons from the affected father. And this example is characteristic of vitamin D resistant rickets. Here are some X-linked recessive disorders: Duchenne muscular dystrophy, hemophilia A and B, chronic granulomatous disease, Wiskott-Aldrich syndrome, diabetes insipidus, Lesch-Nyhan syndrome, and Fragile X. The biochemical basis of metabolic disorders. Enzymes are affected. These accumulate substrate or an intermediate. One example is galactosemia, where galactose is accumulated. When the enzyme is affected, the product can often disappear, such as albinism causes absence of melanin. Another form of an enzyme affected is where another enzyme is unaltered. For example, cystic fibrosis has retention of neutrophil elastase, which destroys lung tissue. Receptor alterations include familial hypercholesterolemia, where LDL receptors are reduced or lose function. So in this schematic, the substrate causes an intermediate, and then the second enzyme can create another intermediate, and then there's a product, and you can have the substrate converted to an end product and a mirror pathway, and you get feedback mechanism. And the intermediate buildup from intermediate 2 can cause a series of other end products. Protein structural changes include thalassemia, where the hemoglobin variants have different synthesis rates forming. There are also changes in drug reactions. So the malaria drug primaquine causes hemolysis in cases where there is glucose 6-phosphate dehydrogenase. Structural protein defects include Marfan syndrome, which has defects in fibrillin affecting deposition of extracellular matrix. Ehlers-Danlos syndrome is another example, and these produce weak elastic structures in the aorta and fingers and collagen deposition in the joints. With the increased cholesterol and familial hypercholesterolemia, intracellular cholesterol inhibits 3-hydroxyl-3-methylglutarate coenzyme A reductase. This is HMG-CoA reductase, which synthesizes cholesterol. This enzyme is inhibited by statins. Cholesterol activates acyl coenzyme A cholesterol, which stores cholesterol, and cholesterol suppresses LDL receptors, which reduces cholesterol accumulation. Lysosomal storage disease is another broad category, and you have hydrolytic enzyme storage granules involved in metabolism, modification, and degradation. This is a schematic showing some of this as lysosomal enzyme protein, rough endoplasmic reticulum. There's mannose-6-phosphate, mannose-6-phosphate receptor, the Golgi apparatus with the vesicles being formed. Lysosomes break these down, and you get phagocytosis, heterophagy, and then these particles are extruded. Lysosomal defects cause reduction of substrate or accumulation of product or toxic intermediates. These include a spectrum of disparate enzyme deficiencies involving glycogen, such as Pompe's disease, sphingolipids and Tay-Sachs disease, sulfatidoses and Niemann-Pick disease, mucopolysaccharidoses, such as Hurler's disease, and mucolipidoses. Glycogen storage diseases occur with deficiencies of enzymes at various points of glycogen metabolism. The nomenclature is described as type 1, 2, etc. Here's an example of that. So this is a type 4 liver phosphorylase kinase, activates phosphorylase. These are categories of 5 and 6. Debranching enzyme, glucose 6-phosphatase, this is the number 1 category. Phosphofructose kinase is 7. Maltase, and these are involved either inhibiting or accelerating the synthesis and accumulation of these substances. So you can have biological errors at any of these points of these enzymes with the corresponding disease. Polymorphisms, co-inheritance of variant alleles with low penetrance. This is a mode of exclusion of other forms of expression. For example, type 1, type 2 diabetes mellitus, also hair, skin, and eye color depend on co-inheritance of alleles with low penetrance. There are also chromosomal disorders. We are born with 22 pairs of homologous chromosomes and a pair of sex chromosomes, either XX or XY. The nomenclature: a male with trisomy 21 has 47 chromosomes XY and extra 21 chromosome. The short arm is designated as P, the long arm is Q, and mosaicism is where two or more chromosome populations have a different chromosomal complement. So among the various changes you get, you have translocations, where you've got a gene here, a gene there, identical break locations with a balanced reciprocal transformation of the translocation of material. You can also have centric fusion, the break at this point, the break at the other point, and you can get fusion of all of it with a lost smaller component. This is called the Robertsonian change. Isochromosomes can break this way. You can also get deletions with the reannealing of the chromatid with a separate fragment. You can have inversions where these flip. There's a paracentric one where involving a polar end of the chromosome, or you can have pericentric, which goes around the midpoint, and it's a mirrored translocation right here. Also, ring chromosomes can occur, and you can have loss of the end fragments in the process of annealing the intact genome. Trisomy 21 is a meiotic non-disjunction. 95% of cases have a maternal origin for the extra chromosome, less commonly a Robinsonian translocation or a mosaic of chromosomes 46 and 47. A spectrum of disease occurs where you have a mental retardation, abundant neck skin, epicanthic folds and flat facial profile, simian crease in the palm, congenital heart defects, intestinal stenosis, umbilical hernia, predisposition to leukemia, hypotonia, and a gap between the first and second toe. You can also have trisomies. Pei-Tau is trisomy 13, and Edwards syndrome is trisomy 18. Sex chromosomal disorders are more common than autosomal aberrations. These are complicated by inactive of one X chromosome and the presence of Y chromosome genetic material. The Lion hypothesis is where one X chromosome is inactivated, and that implies that males and females have the same amount of X-linked active genes, and that females are mosaics for X chromosomes. In Klinefelter syndrome, you have male hypogonadism, one Y chromosome, and two or more X chromosomes, 90% of which are XXY. There's equal parental male and female non-disjunction of the chromatid. Incomplete Lion inactivation creates effects on males with the disease. You also see abortive inactivation of androgen receptors on the X chromosomes. Turner syndrome is complete or partial monosomy of X chromosomes occurring in one of 2,500 females, complete monosomy in 57%, and this is a 45X karyotype. With partial manifestation, a monosomy, you miss components of the short and or long arm of the chromosome. The remaining X chromosome has a 75% maternal origin. And this is Turner syndrome. There's a low posterior hairline, webbing of the neck, short stature, coarctation or narrowing of the aorta, broad chest, widely spaced nipples, a cubitus valgus where the arm is abnormally bent outward and flails away from the body at rest. Streak ovaries, which are fibrotic ovaries. There's no oogenesis occurring. Infertility, amenorrhea without periods, pigmented nevi, and peripheral lymphedema at birth. Trinucleotide repeat mutations occur. These are additions of trinucleotide repeat sequences which destabilize DNA and impairs function. Among these are Fragile X. This is a group of disorders characterized by trinuclear mutation in familial mental retardation gene in the X chromosome. The second most common cause of mental retardation behind trisomy X21 is X-linked, affects males, rare in females, and occurs by amplification of repeat sequences. So you have a carrier male, normal female. The X chromosome is a premutation. The phenotype is normal. Normal X chromosomes here, normal. And there's a carrier female. X chromosomes, again, normal. There's a premutation right here, normal. This is a normal premutation. These are carrier females here. And you can have unrelated, which gives you normality here. If there's a normal premutation here, you can get abnormal meiosis and recombination with a full mutation with an affected phenotype, and normal full mutation affecting 50% of females, often mildly and normal. So this is Mendelian genetics, where eventually you get from carrier male to affected male and female with a normal full mutation. Genomic imprinting is where one allele from one parent or the other is inactivated during gametogenesis, so one copy is functional. In one disease, Prader-Willi syndrome occurs when the functional allele is inactivated in the paternal chromosome 15 long arm. DNA analysis indications are used for tumor diagnosis, neoplastic clone detection, gene therapy options, treatment efficacy, detection of microorganisms, antimicrobial treatment efficacy, and detection of genetic disease. That's it for this talk. Thank you.